Okay. Can you hear me okay?
Yeah, I can hear you.
That's fine. Hello, Tom. Maybe, hi.
Let's wait for a few more people here. Sure.
Okay.
Hello?
Hello.
Hello. Hi. How are you?
Wait another minute or so. All right. Okay, welcome to the meeting today. Hope everyone had a good week. I was talking to Richard before the meeting, and he said he just finished the paper that he presented last week. So I don't know if he wants me to distribute it to the group at some point, but so we'll still be discussing that, in subsequent meetings, maybe at the end of this meeting, if anyone had any questions or comments that they're thinking about from that presentation. Thanks again for the presentation last week. This week we're going to turn to Tom Portegys, who's been a member of the group for many years, and he's going to present on he's doing a lot of simulations with cellular automata and bio-inspired modeling. And so before we get to that, I wanted to share a paper. I don't know, maybe Tom will talk about this paper in the here's Ujjwal. Hello? It's a new paper on growing cellular automata. So let me share my screen. This just came out recently in a journal called Distill. And for those of you who aren't familiar with Distill, it's a machine learning journal that's, they present things not in PDF format, but they present them in this interactive format. So it's an actual journal where people publish these interactive pieces. So let's talk about this for a couple minutes. So this is a paper called Growing Neural Cellular Automata. The subtitle is Differentiable Models of Morphogenesis. And so this journal, Distill, is actually a machine learning journal. So this is a little bit different than what they usually publish. They usually publish things in more mainstream machine learning, but this is a morphogenetic approach to complexity. And the authors here, we have a couple of authors. Michael Levin is the last author on this paper, and he's actually well known in the developmental biology and morphogenesis community. So this is the paper. I'll put a link to it in the chat. Okay, yeah. Devansh says distill is cool. Yes, it is. Okay. So that's the link. And so this is the paper. So it starts off with this animation. So the way this works is that you can... all right, let's start over. You can set the speed and you can set your target shape. So you can start maybe with a gecko here or a lizard. You can start with a smiley face, a human eyeball, a fish, a Christmas tree, a pretzel. I mean, they're trying to be cute, but they're also trying to cover a lot of different shapes. But why don't we settle on this butterfly? So you can settle on this target morphology. And the idea is that you're going to take this cluster of pixels and you're going to run a simulation that forms this morphology. And so if we go back, we reset it, and it's starting to grow at a very slow speed. We can speed it up a little bit more. So it's at half speed, and you get this butterfly. And it's kind of pixelated, but you can see the process here. So let's start over. Butterfly from a little cluster of pixels. So there are different types of models. So growing, persistent, and regenerating. And so they're described down here, these different types of experiments. But they're all basically very similar in terms of their outcome. So this is
you can disrupt the image with the regenerating one and see what happens.
Okay.
Just swipe across it.
Oh, like this? Oh yeah, okay.
And it recreates itself.
Oh, all right. So yeah.
If you don't do too much disruption.
Yeah. So that's the regenerating. Regenerating actually has like where you can cut it and then it reforms. So how does this work? So this is actually, you can try this in a Colab notebook for those of you who are interested in that. So if you want to download the notebook, it gives you the code and everything. So how does it work? So in the paper they get into like how this works. So you get a description of morphogenesis, and that it's an example of self-organization. So they're using cells as the building blocks of these bodies, and they're using rules, and those rules are very similar to what you find in a cellular automata. And they walk through the model here. So here's the model where they have a number of different steps in the model. They actually use a pretty advanced model, and it's actually more than just a cellular automata. There's a lot of machine learning involved in this as well. A lot of encoding and decoding of things. And so I'm not going to go over that right now, but you can read about that. And then they have a way to determine cell state. They have your cellular automata rules, and they kind of walk through that. And then they have their different experiments that they profile in this article. So learning to grow is experiment 1. Experiment 2 is what persists exists. And then experiment 3 is learning to regenerate. And so they kind of walk through all of this. And there's an experiment 4, rotating the perceptive field. So each of these cells, I guess, are perceiving things in their environment and they're responding to it. So Michael Levin has actually written a lot about decision making in cells, and so he's interested in that area. That kind of feeds into this work as well. So related work, he talks about PDEs, which are partial differential equations, and their role in this process, neural networks and self-organization, and swarm robotics. So this is probably going to be of interest to a lot of people in this group. I would go through it, and maybe if someone wants, unless Tom's going to talk about it in his talk, maybe someone could present it on its own, because it probably deserves that kind of attention. But I just wanted to point that article out. And so that sets up kind of like Tom's talk. So Tom, if you're ready, can you share your screen?
Yep.
Okay.
I think you have to unpresent, right?
Oh yeah. Okay. I'll stop presenting.
Can I select the whole entire screen? Here it is.
Yeah.
Okay. Okay, so thanks for presenting it. Bradley, I was going to mention that, but not in that level of detail, but that's great. But the distill venue looks really cool. I think I'm going to probably submit my honeybee simulation to that and see how it goes. Yeah, so it's got some, it'd be nice to have put something out in front of the Google machine learning people. So, okay. So what I'm going to do is hopefully just kind of do an overview of a couple projects. One is Morphozoic, which Bradley and I and Dick and Steve Grew and Gabriel Pasquale worked on a few years ago, and it's a morphogenesis model. I'm not going to go, I've got a couple demos with it, and I don't want to be too daunting here, but I'm not going to do every one of these slides, so I'm just going to kind of go overview level. So, and then with some demos. So, can everybody see this? Morphozoic, the slide there? Okay, so it's a cellular automaton. It's along the lines, that sort of flavor of what you just saw there, where you have various objects kind of growing into other shapes. So what this does is it builds up a set of rules. It basically, the way it works is you kind of hand code some kind of morphogenesis process, and Morphozoic watches that. And then it develops cellular automaton rules. And then using those rules, it can actually recreate the morphogenesis process. And it can, you can feed those rules into a neural network, artificial neural network, and it can learn it. So you can have a nice, compact, fast version of it, which is also very nice for like dealing with noise and things like that. So we wanted something that had it's a cellular automaton, but it also embodies a scheme that's computationally feasible. What you don't want to have is, with cellular automaton, you can get this explosion of bits if you try and capture too much neighborhood information. So Morphozoic has a way to deal with that. It basically aggregates stuff that's far away in space. So let me go to the picture here, it probably says better. Where's the picture? Okay. We don't have the pyramid picture in here. Okay. So go back up here. So here's the cell has a state. This is standard cellular automaton stuff. It emits, senses, and reacts to signals. Signals carry information about state of neighborhood cells. And a field is the confluence of signals sensed by each cell. And the dog's bothering me here. I wanted to get a picture of this. Okay, here's a picture. So like you might have a shape that looks like this on a grid, and it has different cells in different states. And the way this works is you have basically a pyramid of expanding in space neighborhoods. So you can have an immediate neighborhood, which is like the Moore neighborhood, which is 3 by 3. And then that would then you'd have a bunch of 3x3 neighborhoods in a 9x9 neighborhood, but it's actually 3x3, it's just bigger. And this is the aggregation scheme, so you don't actually get this combinatorial explosion. We're using kind of a histogram of cell type. Each one of these cells has a different state, so you can aggregate those into kind of a histogram, or as I'm thinking of it nowadays, the concept of a centroid vector in each of these sectors here of a bigger, larger neighborhood. So you can stack these neighborhoods, so you get this kind of pyramid. So basically you have a context. So a cell has this, can have an arbitrarily large context, and what that allows it to do is determine what to do. That context is called a morphognostic, and this other thing, a morphognostic and a metamorph is part of a metamorph, and morphognostic tells the metamorph what is the action of the cell. So the cell could do like change state, essentially. And here's what you could do with the, I told you could do machine neural network, artificial neural network. So what you can do is, you put in the neighborhood of a cell, and then you output the action, which is the cell, how the cell should change state. And this turns out to be very handy because these rules you can get a huge amount of rules. So let me do a little demo at this point. I think that's the best thing to do, which I last night located. So a couple of these things you could do is we can do okay, let's do the, as you saw with the thing on Distill, you can have a repair sort of functionality. Let's see what we can do with Morphozoic along the same lines. So this uses this sort of famous or now infamous vision image called the Lena picture, which came from a Playboy, which is now not politically correct, but we used it anyway. We probably will not do that in the future. But so what you got here is kind of like a edge detection sketch of the image, and Morphozoic was taught that given this sketch, you should be able to recreate the image to some degree. So let's see what it does. Oops, that went pretty fast. Okay. So it took the edges as the starting states, and each one of those cells said, oh, what should I do? Look at my neighbors. And over time, it develops, you can kind of see the picture here. It actually recreated the image. Now here's another one, which is Turing. There's a famous, the Turing famous reaction diffusion equations. So let's see what they look like to create patterns. And it's coming up. Takes a while. It's crunching away. And I can hear the fan going on my computer now because it's crunching. Okay. So what this does is it takes the Turing reaction diffusion takes an arbitrarily random bunch of pixels or cell states, and if you apply the reaction diffusion with certain parameters, if you change the parameters, you get different patterns. But so this is one of the parameter settings. So it crunches away, the cells start changing states. And they're sort of coalescing. This is the so-called leopard spot pattern, if you've ever seen this. But you can have lots of different patterns, stripes, all kinds of stuff. All from a pair of equations, differential equations. So the cells are kind of coalescing into a pattern of, oh, you know what? I went just backwards. Okay, so what you're actually seeing here, I wonder why this looked a little crappier than it should have. Okay, so this is actually Morphozoic after it has learned how to do the leopard pattern by watching the Turing reaction and diffusion equations that work. And you can see it comes up with sort of, it will go on and get better and better, although it could get even more better if I play with it some more. But you can see a bunch of dots here, a bunch of spots. So it's learning, it had learned to generate the spotted pattern. So let's see if I can actually show you the real Turing thing that it learned from. That will be this one. So here again, we start with a bunch of random pixel states or cell states. And this should look nicer. Because it's from Turing. Okay, here we go. You see, Morphozoic watched this, and then it did its own thing. It learned rules to build this. All these look nicer. Okay. So here's leopard. Here are the leopard spots. Okay, and that's all I wanted to show you for demos of Morphozoic. So let's see if there's. We've also done a couple of things, like the paper has Conway's Game of Life, cellular generation. Evolution of a very, very rudimentary gas dilation sequence, which is an embryo has this, I'm sure you people know more about this than I am. I'm not a biologist, I'm a AI machine learning person, but I have interest in computational biology. So it's like gas dilation is where you have a embryo and it, like there's this, it has this pushing in of the beginnings of a digestive tract. It's actually more than that, but also it does a demo of neuron pathfinding, which is something you have when brains are developing. They kind of reach out and talk to each other, or so they can talk to each other. And you just saw Turing's reaction diffusion morphogenesis. So Conway's Game of Life, it can watch a game of life doing its thing. It's a famous cellular automaton forming all these kind of figures that do all these interesting things. And it can watch those and figure out what the rules are. So it can recreate that. It can regenerate cells. This is just kind of a beginning sort of deal here. You just saw this with the Lena picture. it can learn to generate a full image from like a rough sketch of the image. But you have to show it what it wants, what to do, and then develops the rules. Here's the gasculation. Super simple. This thing, it starts out as a square and pushes in. I think that's pretty much all it does. And, here's neuron pathfinding, where a neuron will send out axons to target neurons. And it can learn to do that. And here's Turing again. And, okay. So, that's pretty much what I wanted to say about Morphozoic. So that's the technique that it uses. So let's go back to, I want to show you a couple other projects that I worked on after this. The next one is a pufferfish, so let's watch a little movie. This is a pufferfish. Well, there's a certain kind of pufferfish that lives off the coast of Japan, and people found it to do these amazing, build these amazing nests, which look like this thing. So this little critter builds this nest on the seafloor, and that's the male that does this. And it has all this kind of wheel shape. It has a central area which is very fine-grained sediments in there. And David Attenborough thinks this is just the bee's knees here. It's the most amazing thing you've seen. So I thought, I approached this project at a different sort of angle. I want to do animal behavior and not just a morphognosis. The Morphozoic, as you saw it, is kind of like something that's sort of static. It's dynamic, but it's all in space. But animals do things in time. So this behavior, this critter, goes over time. It changes. I mean, the fish is changing location and it's moving on in time. And so I developed this kind of related system called Morphognosis. I like to come up with names, but it has a little bit of a sense. Morphognosis means the shape of knowledge, and I call it the shape of knowledge in space and time. And I think I chose not only time but also space because, as you might be aware, mammals. Well, a lot of animals, mammals are one of them. I don't know about other animals besides mammals, but we have these special structures in our brains called grid cells, and there's other ones called place cells, which are specifically wired to do spatial geometry processing. So we were able to, mammals were able to navigate, take advantage of the physics of the world, that it has a 2 or 3D geometry if you're 2D, if you're on a flat surface, and use that as, so it's this stuff that's wired to be able to leverage the fact that the world is a spatial place, as well as temporal place. Because most neural networks, artificial neural networks, just do temporal, essentially, at least the recurrent neural networks that I'm interested in. So here's kind of a picture of this. So you have a brain, produces behavior, again, kind of like Morphozoic, grabs off rules from the behavior by watching the behavior happen in time. Chucks those rules into a neural network, and you come up with a neural network that produces a behavior. And here's a picture of the event pyramid, which is similar to Morphozoic, but I'm calling this a cone now. Because it's kind of like a light cone sort of thing, where you have things in the past can be farther and farther away. So you have events that are up on the top of the cone. You have things that are recent or present right now, currently present. And events, excuse me, farther away in time can be more extended also in space. They can be, they don't necessarily have to be. So this provides kind of a kind of context for controlling behavior. And once again, you want to do some kind of trick to avoid combinatorial explosion, which I use the aggregation trick. There's got to be some other tricks, which I'm exploring, using multiple centroids, but I haven't done that yet. At some point I will. So here's a couple of equations that describe how these neighborhoods are constructed in space and time. So you can see, here's another picture. Here's the cellular automaton here. Here's a 3x3, and then there's a 3x3 of 3x3s, and then there's a 3x3 of 3x3 of 3x3s. So you get, and here you can see kind of a histogram or a centroid of events, features that have happened in that chunk of space-time. And, okay, and then you do the same thing, you plug this into a neural network and learn it. Nice. You get the concise, you get the speed, you get the fuzzy ability to deal with noise. So let's go watch a demo of this next. Let's see if I have the demo right here. Nope, we'll have to go down. Yeah. So again, it's called Morphognosis. And we're going to go here. And we're going to and this is a crude nest, okay? And when I submitted this for publication, someone else just before that said submitted a very, very nice pufferfish nest. I mean, it makes a really cool looking one, just like the real one. And so I said, that's not a very good nest. Well, I'm not really trying to actually build a nice nest. I'm trying to study the behavior of something that does something interesting. And this nest building is interesting. But the one that was really nice is actually hand-coded. So I'm not interested in that. I'm interested in some kind of generalized learning here. So I should have made that clearer, but eventually got it published, so that's okay. Okay, let's watch the pufferfish do its thing. So there was a hand-coded thing that made the nest. Morphognosis watched this fish build its nest, and then it built up these rules, and then I used a neural network to execute the rules. So let's see what it does. Okay, so this red triangle in the middle is the fish. And the fish can actually see the elevations. So this fish actually wants to kind of scrape out the elevations to get them all looking like that round nest. So you see it sees three cells in front of it, and it's elevation 644, and that's actually what it's looking at. And then it's responding with wait, just sort of sitting here for now. It also uses the previous response. It turns out it's actually good to know what your previous response is to determine what your next response is. So let's have it kind of step along here, and we'll let it go a little bit. So now, see, what it's doing now is kind of plowing out, it's flattening out the area. And so it plows out this nice little central area, which is a square because it's this, grid. And now it's actually sort of, you'll see it kind of looking like, we'll get it going a little faster here. But it has to keep track of all its context, like where it is, when it is, what it's doing. And that's what Morphognosis allows it to do. Okay, here we go. It kind of looks like a nest. This is not the gnome art. This is, I didn't have that handy. I had to learn it. This is fairly intensive learning for a gnome network. But... Okay. And we'll just let you finish up faster. It's not going to go fast. Okay. All right, so it made something that looks kind of had this kind of the nest had this kind of wavy sort of circular thing. So that's my version of the nest on a cellular automata. Okay, so I did that last year, and then this year I'm working on something called again Morphognosis, using another. Now, this is a really interesting behavior, and I'm not saying I'm actually telling anybody about how the honeybees work, but honeybees actually do this thing where they, hang on a second, let me show you. Okay, oh, there's that one. This thing right here, honeybees do this amazing behavior where they go out and they find, you know, they gather nectar and pollen, and I'm just saying nectar, I don't know. I'm just simplifying things. Okay, so they'll find nectar from flowers and they'll bring it back to the hive. And if they know, so once they know where nectar is, they do this dance in the hive called the waggle dance. And the waggle dance has two components. So it tells the other bees which direction the nectar is, and that's relative to the sun. And I'm not doing the sun because we don't have a sun in our cellular automata. So it's just going to do kind of make a mental map, which is more than honeybees actually do. It's more like a mammal thing. But it also does, so the direction is of its dance tells it which direction the bees should go. And then the length of this central waggle thing here tells you how far to go. And then the bees go fly off and find, you know, hopefully find the nectar, and they bring it back. It also does another dance, but I'm interested in just the one kind of behavior. So let's look at a little video here, which is me talking about how this thing works. Okay, so what you're going to see, so we're going to have bees. Here's a hive in the middle. Well, let me just run the video here. Okay, so, and then I want to mention that this task is very hard for a conventional neural network to do, unless it's, has some extra stuff, like morphognosis. So, for example, like a very popular recurrent network is called long short-term memory. But if you try and get a long short-term memory to sort of do the dead reckoning navigation to keep track of where the hive is, so when it does blunder on nectar, it immediately gets lost. I mean, long LSTM networks are really, really powerful, but they can't handle this very well. My research seems to point in that direction, and I've actually run this myself as part of the code. I have built this, and indeed that's the case. So you need some kind of help. You need some kind of animals have this thing where they actually have some weird notion of space, and that's what you need to get this behavior to work right. And just as a general note, this is kind of where I'm at, is animals do these kinds of things that, we just don't seem to explore that well enough. To stay on my soapbox a little bit. They do these fascinating behaviors that just stump artificial neural networks as they come out of the box currently. So we need to work on animal behavior because, we have all these things built into us with, amped up to the nth level. So, anyway, let's do that. Let's do more animal behavior. Okay, so the honeybee, here's the honeybee paper in just an abstract at this point. Describing how it's going to work. So it's basically kind of like the pufferfish, but with this other stuff going on. Okay, and I think it's probably it's on GitHub, and you go to my webpage. You can find this stuff here, including the video. So I think, unless I'm forgetting something, I think that's probably my presentation. So.
Okay. Well, thank you for that. That's very good. And I'm sure we have a lot of questions about what you've been doing. And I especially like the comment about the neural networks being unable to do certain things that, people talk about. Like, there's this debate about how neural networks are like the brain or not like the brain, or how it's, able to generate intelligent behavior. But, these are like very specific things that these models are being trained to do. So, like, people talk about like how artificial intelligence can play Jeopardy and win, but it's a very specific type of task that they're doing. And, there's a lot more to behavior than just like, a narrow set of, like maybe an expert system type approach or some sort of like, pattern recognition approach. So I think that you're bringing that out quite well in terms of like how that, what the shortcomings are of conventional techniques.
Yeah, I think, the real world is just full of surprises. We're just constantly running into things that we didn't know were there, and we deal with them. I mean, animals deal with them in general.
Yeah.
So, and, as you said, conventional neural networks, like they're playing a game of Go. Well, you don't have like a bird land on the board while you play. It's like, oh my gosh, it's a bird.
Yeah.
But animals would deal with that. They go like, oh my gosh, a bird, maybe it's going to eat me. I better run away. But, so they're super powerful in these very narrow, very deep silos, But I think to get to we're going to actually, or whether we should, I don't know, go to artificial general intelligence. We need to go to artificial animal general intelligence first. Because we have all animals and us, we're animals, we have all this flexible behavior, we just deal with the world. So that's kind of my my crusade. So that's what I'm kind of doing this.
Yeah.
And we'll see when I submit this paper. We'll see what they say about that. But
Yeah. So I had a couple questions in the chat, or a couple observations in the chat. So
okay, let's see. Chat. Where's the chat?
but the upper right,
there it is.
there
Okay.
it is.
Yep. Gotcha.
So I think this was Dick Gordon. This was about the growing cellular automata. And he said it contrasts with the cybernetic embryo. So, and I know people here probably have heard a little bit about the cybernetic embryo, but this is a series of 2 papers that Rob Stone has written, one with Richard and one with myself and Tom. And we talk about like cybernetics and how that kind of approach can explain something about morphogenesis in the embryo.
Yeah, well,
So-
can you hear me?
Yes.
Yeah, let me be specific.
Okay.
In the cybernetic embryo, the approach is that the embryo does not have a global direct behavior in a global sense. What it does is it sets up a cybernetic system which has a specific goal, and then when that goal is achieved, it includes the construction of 2 more cybernetic systems. So you end up with a tree of cybernetic systems, which results in the embryo. And that's very different from any scheme which had, like the one that we saw with Levin, which strikes me as being a goal-directed simulation where the final state is already known to the
Yeah, that's
simulation.
exactly right.
Yeah.
That's the way that works.
It is the way it works, yeah. And so that says an egg has in its mind somehow an idea of what it's supposed to be, and it keeps checking its own shape against that, what it has in its mind until it gets there. And I think that's a quite fallacious view of how embryos build themselves.
Yeah.
Yeah, that's why Morphozoic actually doesn't have, it discards that final state. It just has a bunch of rules. So there's rules embedded in the thing.
Yeah,
Yeah.
Morphozoic is trying to extract a set of rules.
Yeah.
That's the cybernetic embryo approach. The rules are very localized in the sense that each system has its own goal point, which it then accomplishes, but at the end of the goal, that includes the construction of the next cybernetic system. And so, in fact, in the model, it constructs a parallel, so you get a bifurcating tree of cybernetic systems.
Interesting.
Yeah, and that's quite different from having an overall global view of where you're trying to go. So it's kind of a hybrid in a way, but with these very localized rules. Now, you can get into specifics, but I don't think that's necessary here. It's just this overall philosophy that you don't. Actually, it's almost analogous to tinkering, but not quite. Even in tinkering, which has been touted as a way that evolution works, the assumption that somehow the organism is aware of a global goal. But in the cybernetic embryo approach, there's no such thing. If the tree of cybernetic systems doesn't produce a viable organism, well, that's the end of it. And the only way you get out of that is through producing lots of eggs which have different either recombinations or mutations. So some of them succeed.
How does one organism follow one branch and another one? What makes another one follow a different branch?
No. One organism has all branches.
Oh, okay.
No. This is the structure of an individual.
Oh, okay.
It's just a construction.
So when it unfolds, it kind of decides to go to one branch or another? No, okay.
No, okay. Let me be explicit then. A little more reality. We'll start, first of all, you get some proliferation to get more than one cell, okay? So you get a few, at least, maybe a couple hundred cells or whatever. You got a pile of cells. Now, what happens is you get a pair of waves that go over the organism. What we call the contraction wave, the expansion wave. Those waves propagate through the cells, through a subset of the cells, and change the cells to a new cell type. When the wave is done, it also sets up the conditions for the initiation within that group of cells of two new waves, which then divide the territory into, so the cells undergo another step of differentiation. Okay, so you're branching, and then you iterate this process over and over. And so you're also proliferating cells, so you've got lots of cells to work with.
Okay.
So that's the general idea. That it's this, each group of cells is defined by a wave that goes through it and sets up then a new pair of waves within its new subset. So you have a bunch of nested subsets of cells, and at each step the cells become different kinds. Okay?
Yeah.
But there is no overall goal. If the process is out of hand and doesn't produce a viable organism, it just dies, and you have to, those that have mutations that are more successful at this will be better. It's analogous, in some sense, evolution to analogous to walking a tightrope. Most fall off the wire, but a few get all the way across and produce a viable organism. And the ones that do are the ones that have got the tricks for little local feedback that they could possibly get all the way across the wire. So it's full of local feedbacks, but not much there may be some, but there's not much in the global feedbacks.
Right, yeah.
Okay?
Yep.
So it's a model that needs a lot of exploration, simulation. It's never been simulated.
What's it called? Cognitive embryo.
Yeah, it's in my. If you have access to my papers, it's in.
Okay. Yeah, I do. I have the.
Okay, if any of you listening don't have access, just send me your email address and I'll give you access.
Yeah.
Okay. Here's my email. Put it in chat. Okay. Now the other thing I had is on the pufferfish. That pufferfish nest looks. You could sell it as a centric diatom.
I know it does, doesn't it?
Yes. Okay. Now, I was thinking while you were showing that, well, why not then try to modify the pufferfish model and see if you can fit various centric diatom patterns?
Yeah, it'd be interesting, and that would be more of a Morphozoic thing. Yeah, but yeah, I think it could. Yeah.
Yeah. It's sort of Morphozoic. Yeah, it's sort of a combination of Morphozoic and the pufferfish.
But you're not having a pufferfish make the diatom, no. Right?
Yeah, well, no, of course, diatoms are much smaller.
Yeah.
They are also made, pufferfish patterns are made of silicon. Oh, actually, no, it's silicon. It's quartz.
Yeah.
But that's crystalline, little grains of crystalline sand. The diatoms are more for silica.
Right.
Obviously, the mechanism isn't the same, but there's something uncannily similar.
Yeah, the patterns are definitely.
Yeah, so you might be able to imitate those patterns. Now, we have a chapter in the book on diatom morphogenesis coming out on diatom buckle patterns. And time course of those. Maybe I'll send that to you. It's done with Janice Pappas and me, and she's a much better mathematician than I am, so it's a little bit steep on the math. But you might see in that a way to take your simulation for the pufferfish and start adapting it to centric diatoms.
Yeah, that would be interesting.
Reduce some of the multiple layers of the sudden shift in the pattern and the multiple, as you go out in radius, is exactly what happens with centric diatoms.
Yeah, and it's kind of along the lines of what you're talking about, this cognitive embryos. Like, once you come up with these these local rules, whatever you want to call them, if you tweak them, does it produce a different pattern or produce just crap, you know?
Yeah, I don't know. No one's done a simulation of cybernetic embryos.
So they're like homeobox, you know, rules, you know.
So it would just increase the amount of work for you by yeah, a general cybernetic embryo approach to simulation and see what it produces.
Yeah.
I have no idea.
Yeah. Yeah, it's interesting to know what are the viable, you know, in that space, what are the viable forms, you know?
Yeah, no.
And how can you jump from one viable form to another viable form without killing everyone off?
That's what do you call it? That's the I'm trying to remember Richard Goldschmidt's term.
Yeah.
Monsters. Whether evolution proceeds, but in a gradual form or in big jumps.
Yeah. Oh, punctuated equilibrium or something?
Yeah. Well, it's sort of punctuated.
Yeah.
Saltationism.
Hopeful monsters, I think they were.
Hopeful monsters.
Okay.
Yeah,
I'll have to look that up.
That's around 1926, Richard Goldschmidt.
I had to look that up.
Yeah.
Hopeful monsters.
He was a very good geneticist for his time, but people did not like the idea. And then Smith, Darwin was a gradualist, which people like to keep hammering anybody who sudden jumps in evolution with. But Darwin, if you look carefully at the one picture in Origin of Species and how he describes it, you'll see that he was completely aware that that evolution can occur at many different levels with sudden jumps. So
he was aware of that.
Oh yeah, he was aware of it. But the idea that he was a gradualist is a myth.
Oh, okay.
Heavily propagated myth.
I never got through the whole thing. The Origin of Species is kind of dry reading, but I tried.
Well, when I was 15, I got sick over Christmas holiday, and that's what I read from cover to cover.
Oh, okay. Wow. That'd be good for you.
I can't say I've read the whole book since, but okay. Anyway, this is fascinating stuff.
Yeah.
Okay.
Okay.
I got to run. I've got another.
Yeah.
Okay. Thanks. Yeah, thanks for your comments.
Okay.
Okay, thanks.
So, bye. So does anyone else have any questions before we wrap it up? We're almost at the top of the hour here. Like I said, I can send papers out, or Tom has a link there that you can.
I put a link in the chat at the bottom are the papers with Morphozoic, pufferfish, and honeybees.
Yeah. And so this is a very interesting topic. Yeah. Thanks for the presentations. So we might revisit this in subsequent weeks. If people are interested in talking about it further, please, chat, message me or Tom. We can talk about some other issues related to this. I think it's a good thing to follow up on. Next week we have, I think Jesse's going to have he's going to record his presentation on the free energy principle, and we're going to present it next week, sort of on video.
Okay. That's the Friston.
Yeah, Carl Friston came up with the theory of the brain or something. So Jesse's willing to wade into that.
Okay,
and he's going to put together a pretty good presentation. It looks pretty good, so I'll be enjoying that. And so, but otherwise, have a good week. Remember, we still have spots open for presentations, so if people are interested in presenting, wade right in. Please let me know. Otherwise, have a good week.
Okay. Bye-bye.
All right, thanks. Bye.